Good morning. Let's see. So we are still in the week of October 19th to 22nd. So we're covering neural tissue. So you've got the muscle physiology quiz and mastering, the quiz 3 over chapter 10 is due this evening. Okay. And then Sunday, exam 3 is due over chapters 9 through 11. Okay. All right, so let's pick up kind of where we left off. So we had just begun out in the previous video our discussion of membrane potential. So with resting membrane potential, this is something that happens because the cytosol, remember that's the fluid in the cytoplasm, is going to be different from the extracellular fluid in terms of its ionic composition, the ions that it has and the concentration, as well as its chemical concentrations. Plasma membrane is said to be selectively permeable. Remember that. So certain things can get through, certain things can't. The membrane potential is going to change as it responds to changes in the permeability of the membrane. Okay. Thank you. All right. Yeah, so as I mentioned, the resting membrane potential happens because the concentration or composition of the cytosol is different from that in the extracellular fluid. Plasma membrane is said to be selectively permeable, so some things can get through, some things can't. So as the membrane changes in its permeability, the membrane potential is going to change. And this is going to result because certain membrane channels open and certain membrane channels close. And this happens because those channels are responding to different stimuli. So the primary determinants or contributors to membrane potential are sodium and potassium ions. So these sodium and potassium channels for those ions are either passive or active, okay? So if the ion channels are passive, we call those leaky, okay? These are always open, they're open all the time, but the permeability of those channels can change based on the conditions around it. You also have active ion channels, okay? And if they're active, they're considered to be gated. And they open and close based on the stimuli that's there, the presence or absence of a stimulus. But at resting membrane potential, most of these gated ion channels are going to be closed. Okay. So the types of active channels, so we talked about passive channels, we talked about active channels. So under active, we've got 3 different kinds. So they can be chemically gated, voltage gated, or mechanically gated. Okay. Those are 3 different types of active channels or gated ion channels. Let's talk about the first category, the chemically gated ion channels. These are also known as ligand-gated ion channels. So if you see the word ligand, we're talking about binding of things together, chemicals together. So if certain chemicals, like the neurotransmitter acetylcholine, binds to this ion channel, it can open that channel. So these ion channels, you'll see them on the cell body of a neuron. You'll also see them on the dendrites of the neuron, which should make sense because those are the areas where you can cause something to happen inside the neuron. Because remember, the dendrites are sending a signal to the cell body, and the cell body basically is going to send a signal into the axon. All right, so having chemically gated ion channels at both of those locations can influence an action potential going through an axon. Second category of gated ion channels or active channels would be the voltage gated ion channels. So these basically are going to have their effect based on what's happening with the membrane potential. If it changes, you get a response. You'll find the voltage gated ion channels in the axons of the neurons, as well as the sarcolemma. Remember, sarco means muscle, or has to do with muscle. Lemma has to do with plasma membrane, or skeletal muscle and cardiac muscle. Okay, and that should make sense because charge or voltage helps you conduct an action potential. So it would make sense that they'd be at the axons or in the plasma membrane of these skeletal and cardiac muscle cells, so that you can promote contraction in those muscle cells. So the gate becomes open when it's stimulated. Okay, so when there's something that happens to stimulate the voltage gated ion channel, it opens. And so you essentially, that voltage gate has essentially two gates. You have an activation gate and an inactivation gate. So the activation gate opens when it's stimulated, and the inactivation gate closes to stop the flow of ions. So there's three possible settings or states you can see for these voltage channels. They can be closed but have the potential to open. They can be open, said to be activated, or they can be closed and incapable of opening, which means they're inactivated. Okay. So you can be closed but able to open if you need to, open or close and not able to open. Okay. And typically it's a cycle, okay? So there's gonna be some period where it's incapable of opening before it can become capable of opening again. Okay, third category, because we've learned about the chemically gated channels, the voltage gated channels. Now we're learning about the mechanically gated channels. Okay. And these respond to distortion or kind of movement of the membrane. So it's mechanical. And where do we find these? Well, in sensory receptors, respond to things like touch, pressure, or vibration. Okay. So that should make sense to you that you'd have these mechanically gated ion channels where they are. So where do we have receptors for things like touch and pressure? You might remember a few chapters ago. Yes, in the corpuscles of in the skin and integumentary system. Okay, very good. So always try to link back to things you've already studied. Okay, we'll make the final exam easier. All right, so here's a picture, chemically gated ion channels. Okay, acetylcholine is that chemical that's binding to the channel, makes the channel open. Ions can get through, sodium ions can go through the membrane. Okay, if you run out of acetylcholine, channel closes back up. Voltage gated. So at negative 70 millivolts, activation gate is closed. Negative 60 millivolts, change the voltage, activation gate opens. Okay, if you go to plus 30, if you keep adding positive charge, inside of the cell is going to become more positive. You get to plus 30, and activation gate closes. Okay, and then the ions can no longer get through. Okay. So that's voltage gated. And we'll learn about this in more detail in a little bit, but that's the overview. Mechanically gated, again, normally channels close. If you have some applied pressure or distortion to the plasma membrane, opens up the channel. Okay, get rid of the pressure, channel closes back up. Okay. All right, another concept you need to know about are the graded potentials, also known as local potentials. And before we cover that, I think it would be a good time to stop and talk to your partner about the differences between chemically gated ion channels, voltage gated ion channels, and mechanically gated ion channels. So let's do that now, and you can do that at home as well. Okay, so let's talk about the graded or local potentials. So these happen when you get some change in your membrane potential, but they're localized, so they don't spread far from where the stimulation happens. And any stimulus that opens the gated channels can produce a graded potential. So example here, expose a resting membrane to a chemical. So here, that would cause the chemically gated sodium channels to open because you've been exposed to a chemical. That would make sodium ions enter the cell because of the difference in concentration. That would cause the membrane potential to actually rise, okay? Become more positive. And increase in the membrane potential is known as depolarization, okay? So from here, the sodium ions are moving also parallel to the plasma membrane, so moving along the plasma membrane. And that produces what we call local current. And that can depolarize regions of the plasma membrane near it. So it's graded because it's still in a certain small area. Now the change in potential is really going to be in proportion to the stimulus. So small stimulus, small change in potential, small graded potential. Large or powerful stimulus, you get a more powerful change in potential. Okay, but this also helps with moving charge along an axon or along a muscle fiber. All right, so look at this visually. Again, you have resting membrane potential, negative 70 millivolts. The chemically gated sodium ion channels are closed. Being exposed to a chemical that opens up the sodium ion channels, that's stimulation. Sodium enters the cell. The membrane potential becomes more positive, okay? Now, if you think about it, you're adding positive charge, so the voltage becomes a little bit more positive. Negative 65 is more positive than negative 70, right? Okay. When you start getting spread of sodium ions along the surface. Okay, so these sodium ions start spreading along the surface. Start getting depolarization along these adjacent parts of the plasma membrane. So you start getting a local current, okay, as these sodium ions spread across the inner portion of the plasma membrane. So they're becoming more positive as the sodium ions move away from the initial spot. More terms related to these potentials. So just like an increase in the membrane potential is known as depolarization. If you get rid of the stimulus and the membrane potential goes back to normal, that's repolarization. So then you have hyperpolarization. So just like depolarization makes the membrane potential more positive, hyperpolarization makes the membrane potential more negative. So think about it like the hand signals. So this is resting membrane potential. You have depolarization. All right. Then you have repolarization, hyperpolarization. So this is like our level. Depolarization, repolarization, hyperpolarization. Okay, it's going past the resting membrane potential. And this happens because potassium ion channels open and those ions move out of the cell. Okay, just like sodium moved in, potassium moves out. And then sometimes so much potassium moves out, you go past -70 millivolts. You get hyperpolarization. All right. So these graded potentials, where you see the biggest change in membrane potential is where the stimulation happened, where the stimulus was. And that effect of the stimulus will decrease as you move further and further away from the stimulus. Okay, and this effect of spreading that membrane potential is a passive process. It's those local currents of those sodium ions moving along or moving away from the stimulus on the inside of the plasma membrane that causes that. So a graded change in membrane potential, okay, could involve depolarization or it could involve hyperpolarization because either direction is moving away from the resting membrane potential, right? So if this is, again, your resting membrane potential, removing, making more positive, that's a change from resting membrane potential. Hyperpolarization, moving the opposite direction, is also change in membrane potential from resting. Okay, if the stimulus is stronger, you get a bigger change in membrane potential. Also affects over a larger area if you have a stronger stimulus. All right, so this shows you visually, you measure the membrane potential over time. Again, you're at rest at negative 70 millivolts. You apply a chemical stimulus, could lead to depolarization, increase in the membrane potential. Get rid of the stimulus, you get repolarization. Okay, then you're back to resting membrane potential. A different chemical stimulus is applied, and this time you get hyperpolarization. And then eventually, if you remove the chemical stimulus, you return back to the resting membrane potential. Okay. All right, so graded potentials, okay, so we've learned about them, so why are they important? Well, they trigger functions inside your cells. So exocytosis, remember exo means out, cell, cyto means cell. So remember those vesicles are releasing things out of the cell. So exocytosis of secretions from glands is happening because of a graded potential. Okay, so that's pretty important. And acetylcholine or ACH is going to cause a graded potential at the motor end plate. Okay, so that's the muscle side of the neuromuscular junction. Remember, a neuromuscular junction is just where the neuron and the muscle cell meet up. Okay, so you get a graded potential on the muscle, and that's what leads to your action potential in the muscle, and you get a muscle contraction, right? We learned about that in the previous chapter. All right, so let's take a moment and talk with your partner about graded potentials. You can do this at home as well. Now, we talked about graded potentials. Now, let's talk about action potentials that are related to those, okay? Action potentials are also known as nerve impulses. So, you might get nervous about seeing that term action potential, but really all it is a nerve impulse. So these are changes in membrane potential that are propagated over a distance. Okay, move, like, for example, move along an axon. Okay, so, and they'll affect the entire excitable membrane, so like that whole length of that axon. Okay, so action potentials will start at the initial segment of that axon. Okay, remember that's just after the axon hillock. Learned about that last class, last video. And these actually do not get smaller or don't decrease in effect as you move away from the source. So that's the important distinction between an action potential and a graded potential. So remember, the graded potentials, their effect decreases the further you get away from the stimulus, right? Whereas with an action potential, the effect does not decrease, stays the same. And it's actually stimulated by a graded potential that depolarizes the axial lemma. Remember, that's the axon plasma membrane, to a level that we call threshold. So again, another point of distinction between the two types of potentials. You can have a depolarization, but unless you get the threshold, it's not going to trigger an action potential. Okay, so you can have a graded potential, meaning that you have some variable amount of change in resting membrane potential. But if it doesn't get you to the threshold, no action potential. Okay, so that's the difference between the two. And the threshold for an axon is around -60 to -55 millivolts. Okay. Remember what was resting membrane potential? You might remember what that number was? -70 millivolts. Okay. So you need to increase the membrane potential about 10 to 15 millivolts to get the threshold. Okay, so this is a picture in your book. All right, here's your initial segment. Okay, it's where the action potential is going to start. Remember resting membrane potential. Okay, you get some stimulus, you open up the chemically gated sodium channels. All right, if you get to -60 millivolts, you get an action potential, because it's hit the threshold. Okay, and that makes your plasma membrane much more permeable to sodium, and so you get a lot more sodium coming into your cell at that point. Okay, so you get a huge shift going from -60 millivolts to +10 millivolts. And that whole phase is depolarization. Okay, we'll talk more about that in a minute. All right, another principle here is the all or none principle. So With the all or none principle, that means that regardless of what the stimulus is, if it changes that membrane potential to the point where it gets to threshold, it's going to cause an action potential, okay? If it doesn't get to threshold, it will not cause an action potential. So it's all or none. Either you get the threshold or not. Either you produce an action potential or you don't. And all action potentials are going to be the same. Doesn't matter what the stimulus or how large the stimulus is. All that matters is if it's large enough to get to threshold, which is different from the graded potential, right? With the graded potential, we said the greater the stimulus, the greater the graded potential. But with action potentials, as long as you get to threshold, you get an action potential. Doesn't matter how large it is. Or at least doesn't matter as long as you get to that point. Anything stronger than that, it doesn't matter. It's kind of like if I have a 20-pound weight here on the table and I ask you to pick it up, it doesn't matter whether you're a big person or a small person or, you know, really, really strong. All you got to do is be able to pick it up. You need more strength than that, it doesn't matter. Okay. So action potential is either going to get triggered or it's not going to get triggered. Okay. Either you have an action potential or you don't. That's the all or none principle. All right. Leave one of your short answer question options that you could get. Ask you to write down the steps of an action potential. Okay, so let's get to it. So first step, you get depolarization of that plasma membrane. And it's going to get you to threshold. Okay. Now, once at threshold, these voltage-gated sodium channels are going to open. Okay. Now, another point of distinction, getting the threshold, which type of ion channels were open or which gated channels were open? It wasn't voltage, it was chemically gated. Okay. Now that we have hit threshold, the voltage-gated sodium ion channels are open. Okay. Another influx of sodium ions into the cell, into the cytosol. All right, so we had a negative membrane potential when we started. The inner membrane surface is going to change from being negative relative to the outside to being positive. And you get a very rapid depolarization because of all of that sodium ions rushing in. Okay, so here's a picture of that. Stimulus initiates your action potential. Okay, then your voltage-gated sodium ion channels open. And that happens when you reach threshold. Okay, once those sodium ion channels are activated, makes your plasma membrane more permeable to sodium. Get a rapid influx of sodium ions. Change your membrane potential from negative 60 to positive 10. But wait, there's more. So in step 3, these sodium ion channels get inactivated. Then you start activating your potassium ion channels. So your voltage-gated potassium channels start opening once you get to this positive 30 millivolts. Okay, so your body has sort of an internal checks and balances. You wouldn't want just to have those sodium channels open indefinitely. Okay, so once you get to plus 30 millivolts, those sodium-gated channels close and the voltage-gated potassium channels open. And because the concentration of potassium is greater inside the cell, it's good. The potassium channel is going to make potassium move out. And you get repolarization. Okay? Your membrane potential is trying to get back toward resting membrane potential. All right. Again, here's where the voltage-gated sodium channels are open. Sodium's rushing in, you get to positive 30 millivolts. Then that is the signal that makes the voltage-gated sodium channels close and the voltage-gated potassium channels open. So potassium is going to leave the cell. Resting membrane potential goes back to resting level. You get repolarization. In stage 4, or step 4, you get return to resting membrane potential. Your voltage-gated potassium channels start closing up. Okay, and as you reach normal resting membrane potential, you still have some potassium ions leaving the cell. So your membrane is said to be briefly hyperpolarized. You have a little bit of hyperpolarization because now you're at negative 90 millivolts, right? Which is more negative than negative 70. So, and that happens because most of the voltage-gated potassium channels are closed, but not all of them. Once all of them close, then you get back to resting membrane potential, and that means your action potential has ended. Okay. Okay, so now we're in that repolarization. Your potassium ions leaving the cell, but they continue to leave, and as they start closing, as you get closer to resting membrane potential, a few are left open, so you still get some potassium ions that leave. You get to negative 90, and then you have that brief hyperpolarization. Finally, all the potassium ion channels close, and you get back to resting membrane potential. Yes, ma'am. Yeah. You're welcome. All right, so after all your voltage-gated channels are closed, you're back to resting membrane potential. Okay, no more action potential. Okay, so let's take a break and then talk to your partner about the steps in the generation of an action potential. You can do this at home as well. Okay, so related to the action potential, let's talk about the refractory period. Okay, and this is basically the period that happens between the beginning or the start of that action potential and getting back to resting state. So that whole period is called the refractory period. And during that time, the membrane won't respond to another stimulus like it would normally, okay? Because it's already in, quote unquote, the refractory period. It's already going through the depolarization, repolarization cycle, okay? So it's not going to respond the way it would if it was at rest. Okay, now related to that, you have the absolute refractory period. And during that time, all the voltage-gated sodium channels are open, or what we call inactivated. And the membrane can't respond to further stimulation because, frankly, those voltage-gated sodium channels are already open. So there's not really anything you can do to open them more. They're already open. So any other stimulation is basically irrelevant at that point. Now, the relative refractory period happens when your sodium channels go back to resting state. And it will continue until the membrane potential gets stable. And it has to be a strong stimulus to initiate another action potential. So here, this is relative because technically, even though those sodium channels are closed, okay, look, earlier this morning, or during this video, we talked about 3 states of voltage-gated channels, right? Or 3 states of ion channels. They could be closed and capable of opening, open, or closed and incapable of opening. Okay, so if these sodium channels are closed but capable of opening, it's going to take a stronger stimulus to make them open again. Okay, so this allows them to have another action potential before the whole process of repolarization and hyperpolarization have occurred. Okay. But just normal stimulus wouldn't work for that. Okay, it'd have to be greater than normal. All right, so here's a visual of that. Here's your resting membrane potential. You have a graded depolarization, gets you to threshold. Okay, then you get your voltage-gated sodium channels opening. Here, positive 30 millivolts, sodium channels closed, voltage-gated potassium channels open. Start repolarization. But during this time, till you get to about negative 40 millivolts, membrane can't respond to further stimulation. Okay, so those sodium channels are closed and incapable of opening at this point. Okay, once you get to about negative 40 millivolts, those sodium channels are closed but capable of opening. All right, and so the potassium channels closed. Those sodium potassium channels are returning to their normal states. Okay, so it needs a larger than normal stimulus to get another action potential. So to summarize this again, depolarization happens because you get a rush of sodium ions coming into the cell or into the axon. Repolarization happens because you're losing lots of potassium out of the cell, out of the axon. So depolarization, sodium in. Repolarization, potassium out. Okay, so last video we talked a little bit about the sodium potassium exchange pump. And it helps because it helps us get back to normal. Okay, so it returns concentrations back to their stimulation or back to their levels before stimulation. And helps us maintain our sodium potassium concentrations over time. So this is happening at the same time you're getting like repolarization. This is also happening, okay? but this is an active process. So it's going to require 1 molecule of ATP, adenosine triphosphate, for every time you exchange 2 potassium ions that are outside the cell for 3 sodiums that are inside the cell. And again, this is to bring things back to normal, because remember, in this process, we had a rush in depolarization, a rush of sodium into the cell. So the sodium potassium pump is going to take those intercellular sodium ions and move them out. In repolarization, we had a rush of potassium ions leaving the cell. So we have a lot of extracellular potassium. Okay, and it's going to, sodium potassium pump is going to take that extracellular potassium and bring it back in. Okay, let's talk about propagation. So propagation is just movement of the action potential. Okay, it's moving along the axon. And propagation can be continuous or it can be saltatory. Now, for continuous propagation, this has to do with unmyelinated axons. So this happens in your gray matter, right? Unmyelinated axons. And it will affect one segment of an axon at a time. So here your action potential. Again, it's going to start at the initial segment. You get depolarization to positive 30 millivolts. You get a local current, causes a depolarization in the next segment. Okay, move segment at a time. All right, so this is a visual of that. So as the action potential develops, now at the initial segment here, membrane potential will depolarize, positive 30 millivolts. Local current develops as you get these ions moving down the membrane, spreading away from the sites. You get a graded depolarization, brings axon membrane, your axolemma, to threshold in segment 2. And it continues to repeat segment at a time. In step 3, action potential happens at a second segment. Your initial segment, where you started your depolarization, is now repolarizing. And you get your local current, it's depolarizing the next segment. So you get repeats, repeats, and repeats, and action potential continues to travel down the axon in one direction at a rate of 1 meter per second. Okay, so here's that third step. Action potential happening in the second segment, repolarization happening in the first segment. Okay, continue to move sodium ions, creating a local current. You're going to get depolarization in the third segment. Okay, so that's continuous propagation. Let's talk about saltatory propagation. This happens in myelinated axons, so it happens a lot in the white matter. It's very fast, faster than continuous propagation. It also requires less energy. So the myelin actually opposes continuous propagation. You're going to get local current that's essentially going to jump from node to node. Depolarization happens at the nodes only, and not at the inner nodes. It happens at the nodes. Okay, so here are your myelinated internodes. Get a action potential at the initial segment. You get a local current. You get a graded depolarization in the second segment. Brings the membrane to threshold. Okay, you get an action potential, goes to, you get depolarization to positive 30 millivolts. And you continue to get this local current. Now your third segment is reaching threshold. Okay, but you didn't have to depolarize this whole point, just these nodes. So it's faster. So the diameter of the axon will affect propagation speed of the action potentials. So if you have a larger diameter, so if it's thicker, you get less resistance and faster speed. Okay, so let's stop here, take a break, and talk with your partners about continuous propagation, saltatory propagation, and properties of action potentials. Yes. All right, so there's step one. Step two. So there's actually different kinds of axons. And we can separate them out or classify them based on their diameter, how much myelin they have, and how fast they propagate an action potential. So this one is pretty easy to remember the names of them, just types A, B, and C. Types type A fibers, type B fibers, type C fibers. So for the type A fibers, they have myelin, large in diameter, and they propagate their signals very quickly, 120 meters per second. And they can send sensory information to the central nervous system about position and balance. And they can send motor impulses to the skeletal muscles. So our type B fibers also have myelin, but they're medium in diameter or intermediate. And then they transmit information at intermediate speeds, 18 meters per second. Remember we said the width or the diameter affects the propagation speed. So it's smaller in diameter than the type A fibers, so they don't propagate as fast. But at least type A and B have myelin, so they're going a lot faster than type C, which is unmyelinated and small in diameter. And so they transmit information the slowest at 1 meter per second. That's most of your sensory information comes through type C fibers. So in a sense, the information that's carried in your nerves is routed through kind of like a switchboard. I know, you may not be old enough to know what a switchboard is, but they used to be when people made calls, people that take cords and they'd plug them in, and that would allow calls to be transmitted between locations. And so these nerve signals are routed according to priority. Okay, so critical information gets sent through the type A fibers. So sensory information about things that could keep you alive. Okay, that's pretty important. So that's type A. Also, motor commands that keep you from injuring yourself. Okay, that's also going to be transmitted through type A fibers. Okay. Okay, so we talked a lot about action potentials. Remember, those action potentials get propagated down the axon. They're going to get to the axon terminal and reach the point called the synapse, right? And so it's a specialized place or location where that neuron is going to communicate with some other cell. Might be a neuron, might be a muscle, might be some other part of the body, okay? But that's what a synapse is. Now, if this is a synapse involving two neurons, the neuron that sends the message is presynaptic. It's a presynaptic neuron. The neuron that receives the information is a postsynaptic neuron. It's on the other side of the synapse. Okay. Okay, it sounds fancy, but it's really not once you just think about what the words mean. All right. You have different kinds of synapses. They can be chemical or electrical. For electrical, there's some direct physical contact between the cells. If they're chemical, there's a gap between the cells and the signal gets transmitted via a neurotransmitter. Okay. If you remember in the previous chapter, that's what we saw with acetylcholine. Right? It's being released by exocytosis across the synaptic cleft. So let's start with the electrical synapses. So here we have gap junctions. Remember those from the integumentary system, or cells actually, cells and tissues. So these are connection points between cells, and they lock the presynaptic and postsynaptic membranes together. And because, remember, gap junctions are leaky, you can have ions that pass between the cells through pores, okay? So you have communication between them, the two cells. In the local current, those ions that are along that membrane can affect both cells. You get very rapid propagation of action potentials here. So where do you find these electrical synapses? Well, particularly with some of your special senses like vision. So you have your eye, your brain, you have electrical synapses in the brain as well as what we call ciliary ganglia. Now for chemical synapses, the other type of synapse, these are the ones that are more common. They're much more common than electrical synapses. And they're the only type of synapse that you'll see between neurons and other cells. So you might see electrical synapses between two neurons, but you won't see them between a neuron and some other type of cell. Now, chemical synapses, the cells have a gap between the two, and that gap is called a synaptic cleft. It separates the cells. And just as a reminder, the presynaptic cell is what's sending the message. The postsynaptic cell is what's receiving the message. And again, we're using this term cell here because that neuron could be, or the neuron can be communicating with something that's not another neuron, okay? So here's a picture of the synapse. Teledendrian, which connects to your axon terminals. Here's a big mitochondrion for ATP. You have synaptic vesicles that will release a chemical across the synaptic cleft. So this membrane on the neuron is presynaptic. This membrane on this other cell is postsynaptic. All right, so under chemical, so we had electrical synapses, chemical synapses. Under chemical synapses, you can have a neuromuscular junction or a neuroglandular junction. If the junction is neuromuscular, a synapse happens between a neuron and a skeletal muscle cell, right? Neuromuscular. If it's a neuroglandular junction, the synapse is between the neuron and some gland, okay, glandular cell. Okay, neurotransmitters, okay, very important for chemical synapses. So the chemical messengers here are contained inside synaptic vesicles. And those synaptic vesicles are in the axon terminal in the presynaptic cell. They get released into the synaptic cleft, and they're going to bind to receptors on the postsynaptic membrane. Okay, and they cause some reaction, propagating action potential. And these neurotransmitters can be broken down by enzymes so they don't have an effect that's permanent. And it can also be reabsorbed, and they can also be reassembled at the axon terminal. So what do these chemical synapses do? So basically, with the axon terminal, when it's going to release the neurotransmitter is going to bind to the postsynaptic plasma membrane or receptors on the plasma membrane. Causes some local change or in a specific area, some localized change in permeability of the membrane. Also, it's going to lead to some graded potentials. Okay, we learned about that already. Action potential may or may not be generated in the postsynaptic cell. Okay, remember a graded potential doesn't guarantee an action potential, but you got to get the threshold. Depends on how much neurotransmitter is released. Depends on how sensitive that postsynaptic cell is. So do you have enough neurotransmitter? How sensitive is that postsynaptic cell to the neurotransmitter? Okay, if it doesn't need much, a little will do. If it needs a lot, only a lot will do. All right, I'm just going to introduce this topic of cholinergic synapses. So these are a certain type of chemical synapse. They're going to release acetylcholine at the neuromuscular junction. and they involve skeletal muscle fibers. Okay, we learned about that in the last chapter. So you'll also see these synapses in the central nervous system, as well as all the neuron-to-neuron synapses in your peripheral nervous system are cholinergic. So very common. All your neuromuscular and neuroglandular junctions in your parasympathetic division of your autonomic nervous system. Okay, that's your rest and digest division of the autonomic nervous system. Those are all cholinergic synapses. So they're pretty important. You see a lot of them. Okay, so the events of this cholinergic synapse. This is another one of your short answer essay options. So definitely want to know this. So here, your action potential is going to arrive at the axon terminal, causes depolarization at the membrane. Extracellular calcium ions, remember those, are going to enter the axon terminal. It causes exocytosis of acetylcholine. The acetylcholine is going to bind to receptors on your postsynaptic membrane, cause depolarization. And your acetylcholine gets removed from the synaptic cleft by acetylcholinesterase. Okay, it's an enzyme that breaks down acetylcholine into acetates and choline. Okay, so again, here's your action potential arriving at the axon terminal, the presynaptic neuron. Here's your vesicles with ACH, you know. Depolarization of the axon terminal membrane triggers it triggers calcium ions to enter the axon terminal, which trigger the vesicles to release their acetylcholine via exocytosis. The acetylcholine is going to diffuse across the synaptic cleft, bind to the chemically gated sodium channels. Sodium is going to rush into the cell, the postsynaptic cell. You get a graded potential, could potentially get an action potential if you reach threshold. All right, so you get depolarization ending as your acetylcholine is broken down. Okay. And your axon terminal reabsorbs the choline and uses it to resynthesize acetylcholine. Your acetylcholinesterase is breaking down your acetylcholine. Okay, some of that choline is reabsorbed back in the axon terminal. Okay, so we'll cover synaptic delay in the next video. I'd like for you all to go ahead and talk with your partner real quick about the steps in a cholinergic synapse, and then you can do your exit tickets. All right, well, that has been our video for today.